Hello students, I'm actually covering with you the details of DCPIP and the Hill reaction. To understand what's happening in DCPIP and Hill reaction, we basically already have covered that forces occur in two stages. There's a light dependent reaction, there's a light independent reaction, and the light dependent reaction products these include ATP, oxygen, and reduced NADP, whereas the light independent reaction, it is producing sugars. What's the contribution of Robert Hill? Robert Hill was a renowned scientist, and his major contribution is that he actually found out that oxygen is released in photosynthesis, and this oxygen comes from carbon dioxide or water. So he was able to point out that it actually comes from water and not from carbon dioxide. Similarly, he was also able to point out that photosynthesis, it involves two stages. Number one, photolysis of water, and number two, fixation of carbon dioxide to sugar. So photosynthesis occurs in two stages, the light dependent reaction, the light independent reaction. This was highlighted by Robert Hill only. What was Robert Hill's experiment? The Robert Hill experiment was very simple. Now we can say that it's simple, although that's something of nothing less than a scientific marvel. So what he did was he took a test tube, and in the test tube he took chloroplasts isolated in a buffer solution. Since these are chloroplasts and isolated in the buffer solution, the buffer will maintain the pH because it resists minor changes in pH. And chloroplasts, we know chloroplast isolate will appear green. So the contents of the test tube will appear green. What he did, he covered this, or he wrapped it with an aluminum foil so it doesn't get exposed to sunlight. So the contents would appear green. And then he would later on just remove this aluminum foil to irradiate it with light. We can ignore the aluminum foil for now. So yeah, what he did was the contents of the test tube appeared green. What he did, he added a compound called DCPIP. DCPIP is an oxidizing agent and it's blue in color. So he added DCPIP into this test tube. The contents of the test tube, they turned blue. Why did they turn blue? Because DCPIP itself is blue in color. Next, what he did, he just removed that aluminum foil and the light was irradiated onto the test tube. As the light fell on the test tube in the chloroplast, this water photolysis occurred. Water was split into oxygen and hydrogen was released as well. So oxygen evolved and it is escaped, whereas hydrogen, it combined with DCPIP. You know that when hydrogen is gained by a substance, the substance gets reduced. So we have reduced DCPIP being formed, and this oxidizing agent, basically what happens here, it ended up consuming the hydrogen that was released from photolysis of water. The blue color is discharged and the contents of the test tube, once again they turn green. So the blue DCPIP turns colorless. The control for this experiment was the same, that he again took the contents of a test tube in the chloroplast isolated in a buffer solution. They all appeared green. He added DCPIP, but this time DCPIP was again blue, but he placed it in dark. Since the light was not irradiated, water photolysis did not occur. What was the conclusion? The conclusion was that yes, light energy is mandatory for splitting of a water molecule, which releases oxygen, and it releases hydrogens as well, and the hydrogens are then used up to reduce DCPIP. Since here in the dark there is no hydrogen being produced because of photolysis of water, the photolysis of water is not occurring, so the hydrogens are not there, and when hydrogens are not there, DCPIP will not get reduced. So this is what actually Robert Hill contributed. The Hill's reaction can thus be summarized as DCPIP, and again DCPIP. This DCPIP is blue in color, so I'm saying this is an oxidized state. So if you just place it with chloroplasts in light energy, in the presence of light energy, it would turn, DCPIP would turn colorless. So oxidized turns colorless. So oxidized is blue and colorless is the reduced one, because water got split and oxygen got released, and the hydrogens are gained by DCPIP. We already have seen that oxygen comes from water and not from carbon dioxide. We already have seen that the light dependent reaction here is actually identified as a separate reaction in which sugars are not produced at all. Probably you can just test the solution with Benedict's test and you will see that no sugars are produced there. Chloroplasts, they have a reducing power. This was concluded. And chloroplasts have reducing power because they have NADP. NADP is performing the same function as DCPIP in an intact leaf. Water, in the presence of an enzyme and light energy, it turns into 2H plus 2 electron and half water, half oxygen molecule, and this is released, and this is the photolysis of water. So we know that this 2H plus ion, these 2H plus ions are the one that get added to NADP together with 2 electrons, and NADP get reduced. Robert Hill actually used DCPIP just to follow the reaction, because what NADP is doing in a chloroplast in a leaf, in the presence of light energy, The same thing DCPIP did in the experiment that was conducted by Robert Hill. DCPIP stands for dichlorophenol, indophenol, so DCPIP. And this is blue in color when it's in oxidized state. It turns colorless when it gains hydrogen and electrons, yes. That's it from my side. Thank you so much.